Okay, now you're in listen-only mode. Well, I think it's going to be a good day for that. Welcome back to day 2. Really excited about today's program. I know you can already see some familiar faces and names on the screen. I think we're going to have just a fabulous conversation. And I just want to thank you for joining us from the comfort of your own home. Obviously, our conference looks a little bit different this year, but it's been really impressive to see how many people have been able to join us and have been able to listen to some of this after the fact. So thank you again. I also just want to take a quick moment to say thank you to our sponsors, our gold level sponsors that have been helping us support this conference. That includes Alexion, Amylyx, Biogen, Genentech, Mitsubishi Tanabe Pharma America, and Pharma. And truly, without your support, this would not be possible. And in these days, we thank everyone for all of their support, as we know that ALS does not stop, and neither can we. So that's what this conference is all about. Morgan, I am going to turn the floor back to you so we can get back to the fun part of the program.
All right, I'm going to pass it on because I'm not fun at all. I'm going to hand you over now to Yeltbacker. He's the Chief of Mission Strategy for the ALS Association. We're jam-packed, Neil, so let's get it rolling.
Yeah, thank you, Morgan, and thank you everybody for joining us. This is one of, I think, the most exciting parts of what we do is to talk about research. So I wanted to give you a quick overview of our mission, a quick reminder of what we're about. And I actually am having some computer trouble, so I can't see what you're seeing. So I'm going to trust that you can see our mission slide. And our mission is really to help people who have ALS and their families and everyone affected by the disease, and to serve this community and also empower them. And our advocacy program is a big part of this empowerment issue, this empowerment effort. And so that's a vital role for you as participants in this webinar. And what we're trying to do is help people with ALS live better lives. And so we're defining that in our mission statement as a life with more meaning. So you get to engage with the community in a way that's valuable to you, important to you, engage with your family, your community, your church, your neighbors, and so forth. A life with security, both physical and financial security, and a life meaning and purpose as well. Can we go to the next slide? And so this slide should be our vision, which is a world without ALS, and what we're looking to do, what our organization is really focused on, is helping people with ALS to live longer lives, to live better lives for both them and their families, until we can find a way to end ALS, to cure it, and to prevent people from developing it who don't have it already. So our research program is a big component of that. And so I'm happy to turn it over, I believe, to Kuldeep Dave, who's our Vice President for Research, to present our research program. So Kuldeep joined us in 2019. He oversees our research program, and he develops and implements our research vision by helping us set priorities and goals for the research program, our strategy, our funding, our initiatives. He's a pharmacologist by training. He has a PhD in pharmacology and physiology from the Drexel University College of Medicine. He worked in the pharmaceutical industry on drug development for a number of years, and also most recently the Michael J. Fox Foundation for Parkinson's Research, before joining us as head of our research program. Kuldeep?
Hi everyone, next slide please. Thank you for joining us virtually, and I hope what I'm trying to do today is give you a very high-level view of what's happening today in research. But before I do that, let's talk about us a little bit. So next slide, please. So who is the ALS Association? And we are the largest philanthropic ALS funder in the world. And what sets us apart, I think, is 3 things. One, these arrows that you see to the two other mission areas within the association, care services and advocacy. And the strategy and the initiatives that research launches are done through both information received by care services and advocacy, and our program then in turn informs these two mission areas. So we're all working comprehensively, synergistically in the same direction. Now, a little bit more about some of the numbers. We are, last year, we committed about over $21.5 million in about 70 projects. And since the Ice Bucket Challenge, we have committed over $110 million. So that's the second thing that sets us apart, is the magnitude. We have over 170 active research projects spanning 15 different countries. And I think it's important to understand that we are not just a funder of research grants. We fund infrastructure projects, and I'll get into this a little bit later. We fund training and education programs. So for example, we have a program for our postdoctoral fellowships where we want to train and get ready the next generation of scientists for ALS research. And third thing that sets us apart is scope, and I'll get into the scope in the next couple of slides. I think it's the most important thing to understand here is we leverage our funding. We did a study last year where we asked our grantees that due to our funding and the data they were able to get due to our grant, what kind of follow-on funding were they able to get? And what we found was that our $40 million of grant funding resulted in $120 million, a threefold increase in funding from large funders like NIH. And this is really the key to what we do. Next slide. Okay, so I think before I talk about scope, I think it's important to talk to understand the drug development process. So here's a funnel graph, and what you're seeing is how many compounds actually have to get tested and the different phases they move into. So you can see the stages on your left: drug discovery, preclinical development, clinical development, regulatory approval. And I think there are a couple of take-home messages here. One, as you see on the top, you have to start out with 10,000 compounds. And here it says 10,000. I used to work in pharma. I could tell you it's actually 1 million compounds, if not more, that you start with in the drug discovery phase. And it takes years for that to get to compounds that you then test in animals. And then it takes years from that to then get compounds into the clinic, which is represented here in the blue section. And then you start to go through the different phases. Phase 1, which is safety. Phase 2, is it effective in a smaller number of patients and safety? And phase 3, which is looking at effectiveness in a larger population as well as safety. And as these phases go, they get larger and they get more expensive. So I think there are two things to take here. One, how long it takes. It takes about 15 to 18 years from a time that you start a screening program, sometimes to get to that regulatory approval. It takes a lot of money. Phase 1 can cost anywhere between $1 million to $5 million. Phase 2 can cost anywhere between $5 million to $30 million. And phase 3 studies, which are the most expensive ones, can cost upwards of $80 million. And so you are already seeing that this is lengthy, this is costly, this is complex. And the scale of drug development here is just beyond all of us here. And I spoke about our $20 million commitment last year. this is way beyond that for a single compound. And so I think what this slide is telling you is that we have to be smart in where we invest and how early we invest so that we can see funding for these stages as compounds move through these stages. I think it's also important to understand at the stage that 95 to 98% of these compounds fail. And so, you may start out with 10,000 compounds, but you're ending up with one approved. And there is this failure rate because biology is complex. And so the more we can get that funnel on the top, the more we can have programs that are going into this funnel, the better it will be in terms of success rate coming out of that funnel and in the bottom. Next slide, please. And so this is, again, showing you the same thing, but just expanding the map a little bit. This is how normally a drug, how you go from biology to a drug development program to clinical phases and then approval by the FDA. What you see very early on is basic science on your left. This is where we try to understand what's different in ALS, what proteins have changed, what activity of our cells have changed. Once we figure that out, then you have targets that you can develop drugs against, and that's where you get into this preclinical drug development phase that I showed you, in the earlier slide. And then after that, you get into your phase 1, phase 2, phase 3. And the circle here is indicating what the ALS Association funds. We have a fairly huge and comprehensive footprint, as you can see. We fund basic science research. We fund preclinical drug development programs at crucial stages and de-risk their program for ALS. We fund, although very few studies, but we have funded some phase 1 and 2 exceptions, phase 2 studies as well. The other thing that's important to fund is risk and causes of ALS. This is how we try to understand how people get ALS so that it helps us develop prevention approaches to ALS. On the other side of things, and if you hit next, we also fund research in managing ALS for people that have ALS today, whether that's research in assistive technology, whether it's measuring burden for families and caregivers, whether it's access to drug, to healthcare, quality of life issues, natural history studies, and so on. So I hope that you get the sense here that our program is multifaceted, solutions-based, comprehensive, and yet we can't do all of this alone. I just showed you in an earlier slide how complex and the scale of drug development, and this is why partnership is important. And I'll point that out as we go forward in the next slide. Okay, so I talked about that. You want to know what the state of the field is today, and I can tell you it's really exciting. Let's start from the top here in red, robust efforts in understanding biology. As I said earlier, we need to understand what's different in ALS, what has changed, what proteins have changed, what mutations are there in our DNA. What is different that is perhaps giving us some inkling about the underlying biology for ALS? Because once we understand that, then you go to this next stage of getting targets. So I'll give you an example. If a protein is reduced in ALS, then that becomes your target, and your approach would be, let's increase the protein level or the activity of that protein so that we can nullify or mitigate the effects that that protein is having in ALS. And so that's when it becomes targets. And the good news is that we have a diverse set of targets in our arsenal right now. C9orf72 and SOD1 being the genetic targets, but we have GDNF, Tregs, urate, and many others I'll show you as we move forward hitting multiple pathways, whether it's inflammation, autophagy. And this is good because, as I told you, 95 to 98% drugs fail in the clinic. So we want that diversified portfolio so that there are more shots on goal as to what becomes a success. If you move down to the blue stage, not only do we have multiple targets, but we have multiple drug approaches for manipulating that target. So, for example, we have small molecule approaches. These are generally chemical chemistry compounds, small compounds. We have antibody type approaches. We have antisense approaches, stem cells. So again, very different ways to manipulate the targets that we want to go after. If you move one stage down to the gray area, institutional engagement, this is important. We can set all of this up on the left side, but if other nonprofits, if government, if venture capitalists are not all involved and growing in the same direction, we're going to slow down. And the great thing right now is that we are seeing that institutional engagement, that nonprofits are coming together and working on initiatives together. And industry is finding synergistic collaborations with nonprofits, with each other to move projects forward. So if you then move down to the yellow on the right side is clinical trial efficiency. And here the ALS Association led an initiative to work with the FDA so that the FDA can provide guidance to trial sponsors. And this guidance has now been published, and trial sponsors are following them as they design trials for ALS. We'll talk about the Healy platform and NEALS in a minute going forward. Community engagement. Look at you all. You're all here. We have over 900 people today joining this webinar. Thank you for joining. But you're engaged. And you're taking part in whether it's the registry, whether it's our Curly program, ALS Focus, which my colleague Jill will speak about later, and other nonprofits that are also involved in engaging the community. So again, I think this graph hopefully tells you the state of the field is robust. It's good, it's diverse, and we want it this way. We want engagement, we want multiple targets, and we want to have community engaged to take this forward. Next slide. Okay, and this is the clinical trial landscape. And currently there are about 32 interventional and 34 observational studies. And what do I mean by that? Interventional is generally where a drug is being tested. Observational is could be a biomarker type study. And Dr. Benatar will speak about biomarkers in the next slide deck. But really, there are over 60 trials that are recruiting and enrolling patients. And you can see them listed here in phase 1, phase 2, phase 3. Couple of things to note. Again, what you see here is some of these are antibody type approaches like the BIIB100 or tofersen. Some are small molecules like arimoclomol or masitinib. They're hitting different pathways. RNA60 is an antioxidant, and so is urate. You have T regulatory cells that hit inflammation pathway. And so again, multiple targets, multiple pathways. The little stars that you see next to some of the drug names are indicate that either we had funded these studies preclinically or had funded that technology. And again, this is an example that funding in that preclinical stage was crucial to get these drugs to this stage. And so that's an important thing to take away. What's another thing to take away is that not all of these will succeed. And we know from past literature and past results that many of these will fail. We hope that many of these are successful. And so how do we set up a system so that we can help all of these trials And one way to do this is through infrastructure funding. And so the next couple of slides, I'll go through how we work with different consortia to make that happen. So the first one is our partnership with NEALS. This is the Northeast ALS Consortium. This is the largest consortium that provides resources, infrastructure, and knowledge for most of the ALS clinical trials. And we fund the NEALS Consortium's infrastructure. We fund them to train study coordinators, to get sites activated, and this is the way you would go about searching for a clinical trial in your area if you were interested. You could either go to our website to participate in a clinical trial, or you could go directly to the NEALS website or call our colleague Curly at the NEALS Communications and Outreach Office. Next slide. Another way to help is by funding the Healy ALS platform trial. And again, this is, I told you earlier, I showed you a slide on how long it takes. And each time, think about if you're playing a game in a stadium and you have to take the stadium down and then you have to build it again to have the next game played. And then you take it down again and then build it again to have the next game played. Doesn't it just make sense to build a stadium and then have multiple games being played at the same time? And that's what the Healy platform trial is doing. This is really borrowing from the Kansas State area where multiple drugs are tested at the same time, and they share a placebo group. And by sharing this infrastructure and common protocol and sharing of placebo participants, you speed up the trial and cut the cost by a third and cut the time by half. Next slide. And we can't do this alone. We need to have partners, and CDC is a vital partner for us. They run the National ALS Registry, and they collect vital information to uncover what causes ALS. We, as you may have heard yesterday, if you joined our advocacy webinar, advocate for $10 million each year for the ALS Registry. And the registry, if there's one take-home message, this is our best case scenario for finding out risk factors for what causes ALS, because data is collected by the registry that is critical here in understanding what the environmental exposures may have been, what the occupational exposures may have been. And what's great about this registry is that data is openly shared with the ALS research community, and so are the biospecimens that are collected. If you are not part of the registry, please become part of the registry and enroll to make a difference. In the website is below. Next slide. And finally, as I said, we can't do this alone. The scope and the scale of drug development is complex, it's expensive, it's huge. And so here's an example of a Department of Defense program set up called ALS Research Program, which has been funding ALS preclinical drug testing for the last few years. We secured $20 million for their last year's funding, which is doubled from $10 million. And this is the largest funder, probably even larger than us in terms of funding the preclinical therapeutic development piece of it. And very happy to say that we're now advocating that program should be expanded and increased from $20 million to $40 million so that they can support even more earlier stage clinical trials. And the next slide. So I'll end with the last slide here, which is, oh, sorry, go back. As you're thinking about next week and reaching out to your Congress people, these are some of the things you want to keep in mind. Please advocate for research and increase appropriation for the DoD's program. Maintain our appropriation for the CDC's National Registry at $10 million. Increase funding for ALS research at NIH in general. It was $105 million last year. And providing funds to nonprofits during this time of COVID crisis. So that's it from me. And if you go to the next slide, and I think you heard me mention biomarkers, and let me just introduce you to Dr. Michael Benatar. He's a professor of neurology and Walter Bradley Chair in ALS Research at the University of Miami. He's executive director of the University of Miami ALS Center, and also serves as the chief of the neuromuscular division and vice chair of clinical and translational research in the Department of Neurology. He obtained his medical degree at the University of Cape Town in South Africa, and also trained in both basic neuroscience and clinical research methods. He leads an active clinical and translational research program focused on biomarkers, which he'll talk about, and therapy development for ALS. He's a principal investigator for both the Pre-Symptomatic study as well as the CREATE Consortium, a rare disease clinical research consortium focused on ALS and other related neurodegenerative diseases. Dr. Benatar, stage is yours.
Great, Culdip, thank you very much. And let me see if I have the slides. Okay, thank you so much for the technical help, and really thank you to Culdip and to Neil and to the association for the opportunity to talk with you today about biomarkers for ALS therapy development. And Culdip really laid this up well in describing the challenges of bringing a drug from idea and discovery through to an effective therapy. And what I'd like to do over the next 15-20 minutes is tell you how I think biomarkers might be able to help facilitate and accelerate that discovery and development process. So let's start out. What are biomarkers? And so the word biomarker is really an amalgam of two words, biological and marker, and so that's biomarker. And there are a couple of definitions out there which I'll give you, but really they're a little hard to understand, and I think this is a concept best understood through examples. So the NIH, for example, has defined this as a characteristic that is objectively measured and evaluated as an indicator of a normal biological process, pathogenic process, or a pharmacological response to a therapeutic intervention. And I'll come back to some of those elements. The WHO, similarly, the World Health Organization, has described this as a substance, structure, or process that can be measured in the body or its products and influence or predict the incidence or outcomes of disease. And there are lots of words there, but I think if we think about some common everyday examples, things like blood pressure, serum cholesterol, a urinary pregnancy test, to make this really modern and current, COVID-19 antibodies. These are things we hear and talk about. Perhaps we don't think about them as biomarkers, but they really are. Things like blood pressure and serum cholesterol, these tell us about our future risk of heart disease, our future risk of stroke. And they can also tell us if we're responding to a treatment. If we take a medicine to lower blood pressure or a medicine to lower cholesterol, we can measure blood pressure, measure cholesterol, and see whether they've come down. And so these are biomarkers that we're accustomed to thinking about and talking about and using in everyday life. COVID-19 antibodies, for example, have emerged in the current era as a marker or as an indicator that somebody has had a previous COVID-19 infection. And so these become an important marker of prior exposure, irrespective of whether somebody was symptomatic or not. And so biomarkers at some level are complicated, but at some level, they're also as a concept, as an idea, I hope, relatively easy to understand. So what about biomarkers in ALS? And the truth is we've been as a community thinking and talking about these for a long time, and we have a plethora of candidate markers. And I've lumped together some of these here to summarize them. Some are based on biological fluids, things like blood, urine, spinal fluid. Some are based on neuroimaging, things like magnetic resonance imaging or MRI scans. And some are more neurophysiological or electrophysiological that really measure the function of nerves and muscles, which we know are the primary organs affected in this disease. And yet, despite this plethora of candidates, we really haven't had validated biomarkers that are clearly relevant to therapy development. These haven't yet emerged, and I would argue until recently. And part of what I'd like to do is explain or articulate a little bit of what I think some of the reasons are for the slow progress in validating some of these markers, and also share with you some recent data that I think helps us move the field forward and advance therapy development efforts. So why has progress been slow? I think partly one thing we need to think about is that we should make a distinction between discovery and validation. And both of these are difficult, but they're very different. Discovery is finding a new idea, a new potential marker, getting a glimpse into something that might be of value. Validation is taking that new discovery and figuring out exactly how it could help, what we can do with it, how we can measure it, and how can we make it practically helpful in the real world. And I think often as a field, we've focused a lot on discovery, perhaps with less attention to validation, and I would argue that both are important. But I think another reason why we've struggled to validate biomarkers and really bring them to the forefront of helping with therapy development is I think we've thought about them sort of in a fuzzy way as a single entity. And we haven't really appreciated there's a nuance to these. And the nuance is that we have to think about them in terms of what the FDA has called a context of use. And what's meant by a context of use is really the application. What's the way in which we hope to use this biomarker? What do we want it to tell us? What's its application? And I'm going to touch on 4 of these ideas today. The one is a diagnostic marker, and I'll talk more about what that means. Then there's a prognostic biomarker, and I'll talk about that too. And then there's a predictive marker, which sounds like prognostic, but it's a little bit different, and we'll go into that. And then there's potentially the most important, which is a potential pharmacodynamic marker, or a marker that would tell us that there's been a response to a therapy. And I think one of the key things is that we have to understand this nuance and this potential different application, because what we ask a biomarker to do and how we go about validating it really depends on what application we think that biomarker is likely to have. So let's start then with diagnostic biomarkers. And the goal with the diagnostic marker is to differentiate somebody who has ALS on the one hand from somebody who doesn't on the other. And so we're interested here, and again, I'm fortunate to be using terminology that's now come into common parlance in the news as we've spoken about COVID-19 antibodies, is sensitivity and specificity. And sensitivity is the ability of the test to correctly identify people who have ALS. So if the test is positive, they have ALS. That's a true positive. On the other hand, we're interested in specificity, and we want to correctly identify those people who don't have ALS. If the test is negative, we want to be confident that they don't have ALS. And so if you look at this schema down below here, we've got the biomarker concentration, and in this example, the green dots, the biomarker concentration is low, and in ALS, the red dots, the biomarker concentration is high. And you can see there's no overlap between the groups. So everybody with a biomarker above a certain level has ALS, and everybody with a biomarker below a certain level doesn't. And so this is a test that we can plot out this relationship, and we can see how well this test performs. And this is a test, a theoretical one, that would perform extraordinarily well. The reality is, well, before I come to that, the best example I can give you of this is electromyography, or EMG. And this is a test that's currently used as an ALS diagnostic, and it helps us decide when somebody presents with weakness and progressive painless weakness, is this due to ALS or is this due to something different, some sort of mimic. I think the reality is that this theoretical schema rarely exists. Usually there's much more overlap between the biomarker in people with ALS and people who don't. And so it's much harder to get that clear separation. And part of the other challenge is that very often tests of diagnostic markers compare people to ALS to healthy controls. And the truth is we have no real difficulty telling those 2 types of people apart. And so right now, I think that the diagnostic markers are not the area where we need to really double down and invest our efforts. By the time people who may have ALS come to the attention of an ALS neurologist and the diagnosis is thought about, the diagnosis is usually very clear. But as we shorten that diagnostic delay and we start to see people with symptoms much earlier, these may become a much greater need. So that's diagnostic markers. Let's talk about predictive markers. So predictive markers are those that help to identify a subset of ALS patients who are most likely to respond to a particular therapy. And why this matters is we increasingly think that ALS is not one disease. It has many different causes and many different biologies, and the treatment for one group of patients may be different to the treatment for another group of patients. And so the best example I can give you of this in the current world is in the genetic realm. And so we know, for example, that if somebody has, let's say, a mutation in the SOD1 gene, one of the common genetic causes of disease, that tells us that this is a person who is most likely to respond to something like a gene therapy that targets that SOD1 mutation. So in this context, that genetic mutation serves as a predictive marker to tell us who are the people with ALS who are most likely to respond to a particular therapy. And so we currently have these in the genetic arena. We currently really lack these sorts of biomarker in the non-genetic arena, but we urgently need these. We need to know who are the patients who will respond to an anti-inflammatory agent, to an antioxidant, to some other form of therapy. We need those with great urgency. So what about prognostic markers? These are markers that help to predict the future course of disease. Tell us, is somebody going to progress quickly? Is somebody going to progress slowly? And we know that there's enormous variability between people. This is a plot just showing the ALS-FRS, the rate of decline of the functional rating scale we use, people declining very quickly, people declining very slowly. And this is very, very variable. And the idea of a prognostic marker is that we might measure this when we first see somebody, and it would predict what does their future course of disease look like. And I've schematized this here: a high concentration of a marker, a low concentration of a marker. And the idea is if we had these markers and we could predict that future, then we would be able to take this variability into account and make clinical trials more efficient. And some potential examples here are neurofilaments, and we'll talk more about those. Urinary P75, I won't have time to touch on that today. But critically important is that a useful prognostic marker has to add value to what we can tell simply at the bedside, for example, based on age, gender, where the disease began, and how quickly it seems to be progressing up to the point of when we first see someone. So what about pharmacodynamic markers? Pharmacodynamic is a fancy word. Pharmaco is drug, pharmacology, dynamic is changing. This is something that would show us that a therapy has actually had a biological effect. And so to help understand this, let's look at two potential scenarios. Here's a scenario in which the biomarker concentration is elevated. It's high in people with ALS, but it's stable over time. It doesn't change as disease progresses. And here's a biomarker that's dynamic. It's progressing as disease unfolds. And either of these could potentially be pharmacodynamic. If we gave an experimental therapy and the biomarker level came down, shown here in green, or instead of going up, it plateaued or perhaps came down, that would tell us, without waiting to see whether there's an effect on function or an effect on survival, whether that drug is having the effect we intend. And this could potentially have enormous savings in terms of the number of people who would need to participate in a clinical trial, and help us make really good, efficient decisions about when to move from phase 2 into those very big, expensive phase 3 clinical trials. And again, neurofilament is a good example of this, and I'll show some data around this in a moment. But just to point out that relevant to neurofilament, there's some very promising experience from a related disease called spinal muscular atrophy that gives us reason to believe that neurofilament would be or should be responsive to a therapy in ALS. So what do we know about these things called neurofilaments? These are major structural components of nerve cells. They're the scaffolding of these things called axons, and they're released into the spinal fluid and also into the blood in response to nerve injury or neurodegeneration. And we can now measure these very reliably using modern day technology. And so I think neurofilament has application both as a prognostic marker and potentially pharmacodynamic. So this is a recent study from the CREATE Consortium, from its phenotype-genotype biomarker study, and looking at both two forms of neurofilament, light on the one hand and heavy or phosphorylated neurofilament heavy. And what we see is that neurofilament light but not heavy really does add a lot of prognostic value, and it does this over and above those typical clinical predictors. And so we think this will be useful in clinical trials. Potentially even more important is the potential pharmacodynamic utility. And what we saw here in the same study, and here we're plotting the levels of neurofilament light, that's this axis, and this is time. So this is light, and this is heavy. And what we see is there's lots of variability between individual people, but for each person, there's a very stable and flat longitudinal trajectory. And so we can see that neurofilament, especially light, doesn't vary that much between people, and it's very stable over time. And so if you think back to that example I showed you, if we were to intervene with a therapy and we could bring that level down, that would show us that the treatment was doing what we were hoping and expecting it would do. So I want to just very briefly, before I close, touch on the potential value of neurofilaments to pre-symptomatic ALS. And so for those who haven't thought about this before, that's an unusual idea, but it builds off of the idea that neurodegeneration in ALS may be beginning well before symptoms actually appear. And so raises the question of, when does ALS begin? And so to answer this question, back in 2007, we initiated the pre-symptomatic familial ALS, or pre-FAL study. This is a longitudinal natural history and biomarker study of people who are at genetic risk for ALS. And I should emphasize, currently we can only do this kind of study in people who are at genetic risk. And we enroll people who are a carrier of any ALS-causing gene mutation. And the goal really is to identify biomarkers of pre-symptomatic disease and potential predictors of the emergence of symptomatic disease, or ALS, and to prepare for a future disease prevention trial. And so here's what neurofilament looks like in healthy controls. It remains low and stable over time, and the gray bar is showing us the maximum value that we see in controls. In people who are at genetic risk for disease but don't go on to develop disease, levels remain low, a little bit higher than amongst controls, but remain low. As we've seen before, people with ALS, the levels are elevated above controls, above this gray bar, but stable over time. And in phenoconverters, these are people who are at genetic risk and they've gone on to develop ALS. We see the levels of neurofilament rise in advance of this bar here, which is time 0, which is the development of ALS. And if I show that to you blown up on a larger scale, here's time 0, ALS. Here's a year prior to ALS. The gray is the maximum level we see in healthy people. And you can see that neurofilament levels increase within the year preceding the development of disease. And so we think this tells us that neurofilament in people at genetic risk for ALS has both prognostic and potential pharmacodynamic utility, predicting when disease will develop, but if we could knock down those levels, tell us that a drug is working and potentially staying off ALS. So let me wrap up then. Biomarkers, I think, are critically important to ALS therapy development, but it's really important that we think about their context of use, and I've spoken about several of these, and I think that the prognostic and the pharmacodynamic are the ones that are most going to be relevant right now, but we have an urgent need for predictive markers in non-genetic ALS. And let's not forget about validation in addition to new discovery. And also to say that I think that these biomarkers are going to empower disease prevention trials in people who are at genetic risk for ALS. And so I know this is being recorded. These slides will be available for those who are interested. Here are the references that I've referred to. And lastly, just very quickly, some acknowledgements. I just want to thank our patients, study participants, family members, all of the people on this webinar who participated in these studies and many others over the years. My colleagues and collaborators around the country for many discussions and helpfulness in shaping these ideas. A special mention to my colleague Joanne Wu here at the University of Miami, who's the person with whom I work most closely and without whom this work could not have been done. My team at the University of Miami, the broader group that we work with through the CREATE Consortium, as well as all of our funding agencies who's so crucial to all of this work. So I'm a few minutes over, not too much, but thank you very much and looking forward to questions.
Okay, Dr. Benatar, thank you. Dr. Deve, thank you. Lots and lots of questions. Thank you all for keeping them rolling in. You have obviously found the question bar where you can submit. Please do, we're going to get in as many as we can. So let me start with a couple of questions about COVID-19 and the environment that the COVID-19 pandemic has created at the FDA and towards ALS research generally. Dr. Benatar, can you talk a little bit about the impact of COVID-19 on the current state of clinical trials?
Sure. So I think, as one might imagine, COVID-19 has posed enormous challenges, not least of which arise from the fact that it's been hard for people who are participating in clinical trials to come to sites for traditional study visits. And those who've been in a trial or a study know what this looks like. You come, you need an evaluation, a measure of outcome, how are you doing, but also very importantly, measures of safety. often blood work, EKGs, other measures to know that the drug is not causing harmful effects. And so what's had to happen very quickly is ongoing studies have had to figure out ways to do most of this remotely. We're fortunate some of the measures like the ALS functional rating scale are well validated for telephone administration. We can do that. But getting blood work, for example, has proven to be a little bit more complicated, and depending on the phase of the study, that may be more or less important. Spirometry, testing respiratory function and vital capacity, has been particularly challenging. That's a high-risk procedure in the era of COVID-19 because it produces droplets and potential transmission of infection. And that's an important measure that we use in clinical trials. And so I think this has posed many logistical challenges to seeing people, to doing evaluations of efficacy, of safety, also logistics of getting study drug to people and being sure they can remain on study drug and remain safe. This has been doable, I think, for oral agents. For drugs that are administered intravenously or intrathecally, those study visits have had to continue in person with much greater caution and appropriate social distancing and other public health measures in place. And I think funding agencies and the FDA have been extraordinarily understanding of the need to make these modifications and to help the research machinery move forward. So I'll pause there.
Okay. Related, and this is for both of you, there are quite a few questions about the process by which the COVID vaccine trials seem to be speeding forward. And why is that the exception and not the rule? Why isn't ALS research moving and the clinical trial process moving as quickly as the COVID vaccine trials? What's the difference?
Who wants to start? Should I start, Calder?
Yes, please.
I guess one thing I would say is to get a cure and effective treatment, we need to know the cause. And one of the huge benefits of COVID-19 or things that empower us to look for effective therapies is we know the cause. That's one of the reasons, for example, in ALS, there's been as much work as there has on the genetics, because if we know the cause and it's tangible, we can go after it. And that's one of the things that I would say is to highlight why research into the cause of ALS or the causes is absolutely critical.
And I would just like to add to that, this is exactly goes to the point I made earlier, why understanding the biology and the underlying mechanisms that are changing is important. We know the genome of the COVID virus, but you know, understanding the causes and mechanisms that underlie the pathology in ALS, I think that will guide us for drug development.
Okay, another COVID-related, sorry, I muted myself. Another COVID-related question that somebody's asked, is there anything in the COVID-19 vaccine trials that could be potentially helpful or inform ALS research?
Yeah, that's a tough one. I don't think anything comes to mind that obviously to me. I do think the overall understanding of dealing with a pandemic that forces people to be isolated gives us, I think, greater sympathy for people with ALS, and that's important. I also think that maybe we're going to learn something about how to help the FDA figure out manufacturing a little bit faster. And there may be other things that we can learn. We're definitely keeping our eyes and ears open through this process.
I guess one thing I might add, Neil, is I think we're still learning what the impact of this virus may be on people with ALS, not just in terms of access to care, but does it have any impact on disease? Certainly, we don't want anybody who's got breathing muscle weakness to develop pneumonia. And I think we're currently in the process of trying to figure some of that out. I will say that through initiative of the NIH, there's a survey currently ongoing. We can find a way to distribute that information to understand the impact of COVID-19 on people who are living with ALS. So to help us learn more about that, please look for that and participate.
Great. And Caleb, I have one for you. given the impact of COVID-19 generally on the state of research beyond clinical trials, what is the ALS Association doing to mitigate the slowing and the difficulties that some of our researchers are having in the labs or getting to labs?
Yeah, good question. With institutions closed, labs are closed, and people can't get in to do research. What the ALS Association did is we reached out to our 170 grantees that I mentioned earlier proactively and asked them about how COVID was impacting, how closing of their labs was impacting their research. I didn't get a chance to talk about this, but we provide milestone-based payments, and researchers have to meet certain milestones before we release the next payment on their grant, and how these closures were going to impact them being able to achieve those milestones. So we're working with our researchers in developing timelines and more realistic timelines to be able to meet those milestones. And so the other thing that we did, and you may have heard about this a little bit yesterday, is our advocacy group led an initiative with over 100 different organizations to advocate for funding in the next stimulus for this type of bridge funding, because as researchers will come back from COVID, their labs are going to need support for staff, for doing the actual research, and ALS Association led the fight, or is leading the fight, to get that kind of bridge funding for our researchers.
The other part of that is most of the folks we fund also have funding from NIH or the VA or the DoD, and we're hoping that the government will continue to provide funding for these labs while they're closed, so people don't have to drop out of research or go into other places. And so part of what we're asking you to advocate for when you go to Congress is that extra funding for these research programs, which will help maintain continuity of research in these facilities.
Okay, thank you. A couple of questions about the Healy platform trials. The first question is, are we funding that trial in any way? And the second one is, can you clarify if that trial is enrolling currently?
Sure, we are funding the Healy trial. You know, the phrase I like to use is rising tide lifts all boats. And the Healy trial is really that concept. By us funding the Healy trial, we funded them for $3 million, $1 million each for 3 years. The concept is that the Healy trial will start with 5 regimens to be tested against placebo, and then as time goes, they will add more regimens to their testing paradigm. And this really helps with, again, as I said earlier, with time and cost, because you're using the existing infrastructure to build and add regimens or subtract regimens if they're not working, and really saving the time it takes to sort of on and off each trial. So this is, yes, we are funding it. We are happy to report that they have, you know, even during the COVID pandemic time, have kept on making sure that their infrastructure is set up, their sites are being activated, their IRBs are being fulfilled and confirmed. So they are continuing the administrative and operational path so that in the event when the COVID pandemic is over, hopefully that they can start and accelerate patient enrollment. And
just to add that it is not yet open for enrollment. I think that was the second part of the question.
Right.
Are you a site of the platform trial?
Yeah.
Yeah.
A couple more questions more generically about people considering enrolling in trials. Someone in our audience asked for advice, somebody in her life with ALS, is trepidatious about enrolling in a trial out of fear that he or she will be placed in a placebo group. What advice do you give to somebody who knows that there's a likelihood that you could be in a placebo group in a trial?
Calder, you want to start or should I?
Michael, you should take this.
Okay.
You're enrolling patients on at your site, and I think you have experience talking about this.
Yeah, so it's a common and an important question, and part of the reason that, well, I think the first step is to understand why it is that we need placebo groups, because I think that's often a struggle. And if you think back to that one slide I had showing the enormous variability in progression between people, if you don't have a placebo group, you really don't know if a treatment works. You know, what I often say to people who ask this question is this is a very personal decision, and so this is a personalized discussion. There's no one size fits all for everybody. But I think that if you don't participate in a clinical trial, there's a 0 chance of getting that drug. And if you do, then there's a 50 or a 60 or a 67 or a 75, depending on the randomization ratio. And many, many studies will have an open label extension, so access to the active compound following completion of a double blind. And so I think those are all things to think about. The other thing to add to that is we always hope that somebody who participates in a clinical trial and receives the active drug will benefit from it. I think when one participates in a clinical trial, it's more an act of helping to advance science, to help move forward the development of therapies without an expectation as to whether one will benefit or not. As you've seen from the history of this, most of the clinical trials we've done have not shown a beneficial effect of the experimental agent. And so we only get to effective therapies by moving the needle forward through research. And so I recognize that, you know, a clinical trial and a placebo is not for everyone, but for those whom it is for, I very much encourage people to participate. I hope that's helpful. There's much more one could say, and I think it's very much a personalized decision with your physician and the person who's talking to you about a trial, and to explore those issues in detail, not just you, but with your family and support and caregivers.
Thanks for answering that. That was very thoughtful. Neil, a question for you about policy and position. We're being asked what the association's position is on early access programs and how we may support those.
So we support early access as a concept. We supported early access policies. I think the early access is great. I think that the challenge comes is if we ever have to make a decision between early access for a handful of people versus advancing science or more resources for more research. And when we have to make those trade-offs, we always favor doing everything we can to get a treatment approved as quickly as possible. Sometimes those are very abstract decisions. As a practical matter, the decision to provide early access to an experimental treatment is always made by the trial sponsor, the manufacturer, and we usually don't have a role in that decision. But we have been working with the FDA to favor open label extensions and think about early access, as Dr. Benatar mentioned, and we got more of that language into the FDA guidance on ALS clinical trials. But in general, early access is great, but faster treatment and faster cure is even better. And where we have to make trade-offs, we always favor speed over early access. And I can appreciate that that's a difficult call. Dr. Benatar, you may have thoughts on that too.
Well, I agree. I think it is always a trade-off, and often I think it becomes a theoretical issue because there isn't access to drug. I think the other thing for perspective is, for all the discussions we've had about open access in the past, none of those drugs have turned out to be effective. And so I think, to your point, Neil, about the critical need to move this engine forward and get to effective therapies, and then we need early and open access to things that are affected. Early access to something that's not isn't helpful.
Okay, gentlemen, I'm going to wrap this up and try to keep us on time this afternoon. Kuldip, may I turn the floor over to you to introduce our colleague, Dr. Yersak.
Sure. Thank you, Dr. Benatar, for joining. So we'll turn the focus, pun intended, onto our ALS focus program. And let me introduce you to my colleague here at the ALS Association, Dr. Jill Yersak. Dr. Yersak is a Vice President of Mission Strategy and has worked for the association for 4.5 years. She helps oversee the strategy projects and programs that span our mission areas, the 3 mission areas that I talked about earlier: research, care, and advocacy. She's sort of the link that links us all together. In this role, she leads our mission programs, including the Roundtable program and the Focus Survey program that she'll just talk about in a minute now. Dr. Yersak earned a BS in biology from Ursinus College in Collegeville, Pennsylvania, and a PhD in molecular cell biology from Thomas Jefferson University in Philadelphia, where I went to school as well. She completed her postdoctoral fellowship in neuroscience at Brown University in Providence, Rhode Island, where she studied ALS animal models. And with that, Jill, the floor is yours.
Thank you, Kuldip. Next slide. So thank you for having me today, and Kuldip, thank you for the nice introduction. I wish I was in person with you. I think like everyone else feels, but it is what it is. So we're going to do the best with what we have. So I'm really excited to be here to talk to you today about a special program that my team and I have built and launched in the past year called ALS Focus. It's a unique survey program whose goal is to better understand the challenges of people with ALS, caregivers, and their families through their disease journey. We cannot know for sure what people are going through we can't understand what people are going through every day unless we ask. And my job at the association is really to help empower people with ALS and their caregivers, both past and present, to share their experiences to help inform change. And I want to thank all the people out there who have already joined our community and participated and signed up for our first ALS Focus survey. And really, without you, I could not do my job. So thank you. And at the association, we know that in order to make change happen, we need strong, robust data. And the survey data we collect is geared on important topics that we all know need improvement. And those things can vary from drug development, clinical trial design, drug payment and reimbursement issues, improving care services and caregiver burden, home health, and relieving financial stress and more. And this data informs programs and policies to make significant advances in these areas, not only at the ALS Association, but also in industry and the government and our partners in other ALS nonprofits. And I'm proud to say that the ALS Focus is designed to collect data and make it open to the entire ALS community free of charge. And this is for researchers, all our industry, pharma partners, all other ALS nonprofits, and people with ALS. This is something we care really deeply about. If we don't share information, we know we will waste time and energy, which is to us is really unacceptable. Another important aspect of the program is its ability to confidentially link other research studies and trials through a code. It's called a global unique identifier, which is like your own personal PIN number. This helps us keep your de-identified information confidential and safe. These global unique identifiers are currently used by the National ALS Registry, clinical trials, and research studies that you may already have participated in. And with this global unique identifier, the survey platform can automatically link your de-identified data from ALS Focus, which is really focused on patient and caregiver burden, to these other studies that I just mentioned. This creates a huge, robust data set that researchers from all over the world can study, again, free of charge, which we're really proud of. And we thank our partners at the Neurological Clinical Research Institute and Mass General Hospital for building our platform for us, and for being great partners and for safely storing the de-identified data at Neurobank. And we can also look at the data by the first 3 digits of your zip code. And this can help us better understand the needs and burdens of people in different areas of the country. We know that people's issues can vary greatly based on where they live in the United States. And this could really help customize solutions to problems based on location. Next slide, please. So most importantly, and what I'm most proud of, is that the ALS Focus is a patient and caregiver run program. I'm just the person who leads the program. I'm not really running it. I am proud to closely work with our Patient and Caregiver Advisory Committee, chaired by my colleagues Ken Menkhaus and John Russo, who will be talking after me, who have been in lockstep with me and my team throughout the development, launch, and progression of the ALS Focus program. This committee informs every decision we make, from the survey topics, survey questions, data sharing, and even more, to move the program forward. And along the way, I'm happy to report we've had a lot of fun, yes, even though it's a survey program, which sounds boring. So together, I'm happy to report that we've made a ton of progress in the last year, with our goal to put around 3 to 4 surveys out a year. In this past February, we launched ALS Focus and put out our first survey on understanding insurance needs and financial burden. This topic was picked and was at the top of our list of the Patient and Caregiver Advisory Committee. We all felt that we needed to know more about what types of insurance people with ALS and their families have, and what are the gaps in the coverage, and how does that impact them and their families, especially financially. This data can help us and our partners in industry and government and other nonprofits inform how we can improve insurance coverage and financial burden on people with ALS and their families. Right now, the first survey is being analyzed, which is really exciting, with our team here at the ALS Association, and we should have the data to share in the next month or so with our community, which we're very excited about. So please stay tuned for that. And in July, we're launching our second survey on what matters most to people with ALS and caregivers. This can help us and our community gain a better understanding of how to improve many different areas involving quality of life of people with ALS and their families. And I also want you to know that all the surveys in ALS Focus are reviewed and approved by an institutional review board called the Western IRB, to make sure we meet all the standards to keep your rights as a participant safeguarded and your data fully confidential. This is something we take extremely seriously. So again, thank you to all of those taking the time to already participate, and we could not have moved forward without you. So I really appreciate your time. So next slide. So enough of me. I want to introduce you to my friends and colleagues, John Russo and Ken Menkhaus, co-chairs of our ALS Focus Patient and Caregiver Advisory Committee, for you to hear directly from them about their experiences working with my team and I on ALS Focus. So first up is John Russo. John Russo, since his diagnosis in 2013, he and his wife Loretta have been advocates for people with ALS at both the federal and state level in New Jersey. There's John. I've known John for years from when I used to volunteer with my home chapter, the Greater Philadelphia chapter. And John has been a consumer reviewer for the Congressionally Directed Medical Research Program at the DoD ALS Research Program for 4 years. John, again, serves as my co-chair of my Patient and Caregiver Advisory Committee. And prior to diagnosis, John worked in the dialysis industry for 29 years in various sales, consulting, and business development roles. John and his wife, Loretta, have been married for 36 years. Also, next slide, I wanted to introduce Ken, and then John can take it away. So, make sure Ken's up. Dr. Menkhaus is a C. Lewis Nelson Professor of Political Science at Davidson College. He specializes in the Horn of Africa and is published in the fields of development, conflict analysis, humanitarian response, peace operations, peace building, state building, and violent extremism. So we can tell already Ken is extremely smart. His work involves both academic research and policy work with the U.S. government, the United Nations, the World Bank, and non-government organizations. In March 2018, Ken was diagnosed with ALS, and since then has redirected some of his research and policy work to ALS advocacy, including service on several ALS Association committees, including recently joining our Board of Trustees, and co-chairing the ALS Focus Patient and Caregiver Advisory Committee with John. So Ken, I'm gonna stop talking, and John, I'd love for you to take it away.
Thank you, Jill. Hello, everyone. Loretta and I are disappointed that we're not all in DC together. This would have been our 7th trip, but we're making do as best we can. We are clearly in unprecedented times. COVID-19 has taught the entire world a lot these past few months. I believe it has taught many to understand what it is like to live the way most of us do, day to day, with a focus on surviving as long as we can. Indeed, COVID-19 has put a greater challenge on our community due to the fact that many of us require outside help from caregivers, and we already struggle financially. Many may feel we're stuck. We have ALS and there is nothing we can do about it. Well, that's of course, unless you are a neuroscientist with a multi-billion-dollar budget and a great idea. There is something we can all do, especially during these unprecedented times. The ALS program is our opportunity to shape the priorities of both the ALS Association and the greater ALS community. Your participation can impact drug development, clinical trial design, regulatory review, drug payment and reimbursement, clinical care, home health, and more. Also, if we patients think about those closest to us, our primary caregivers, I imagine the majority of the time that person is a partner or a spouse. It is difficult at best to transition from partner, husband or wife, to caregiver. It's a huge shift for the primary caregiver to care for a declining loved one and handle all aspects of the life they once shared responsibility for, as well as the new challenges of healthcare, insurance, financial burden. The list goes on. Participation in the Focus program affords caregivers, both past and present, opportunities to be heard. Questions like, what burdens do they endure every day? How do these burdens impact their lives? Can possibly be answered by participation. I believe that what we've shared today is not new news to you. After all, you are the people who participate in advocacy and have already been inspired in some way to help our overall cause. I suggest that we all take stock in our accomplishments over the years, but realize that we still have much work to do. We all learned from COVID-19 how quickly the government and scientific community can rally around a cause. We need to do more to lead the effort to make ALS the cause that science and government make a priority. We can only do that if we gain the support of more people to help share our story. With that in mind, I feel that we need to go out and inspire as many people as we can along our journey. This would include other patients, past and present caregivers, and the general public. Together, let's provide the inspiration to create a world without ALS and join us in the ALS focused community. Thank you. Floor is yours, Ken.
Thank you so much, John. As Jill said, I'm a political scientist by training, so we're hardwired to care about things like representation and voice. Now, there are lots of us out there, both paid staff and volunteers in the ALS Association and in the many other ALS care service and advocacy organizations around the country and around the world, whose mission is to represent and advance the interests of people with ALS and their caregivers. But how do we know what their interests are? How do we know how to advance them? Well, we can think we know. We can think we know because we talk to a lot of people with ALS, or we monitor social media and ALS forums, and those are all helpful, but they're not very systematic, and we run the risk of getting it wrong. And it's certainly the case just because someone like myself or John has ALS, that doesn't necessarily mean we can represent the entire community. There's only one way out of this problem, and that is the systematic, effective use of survey research. Surveys are going to give us the tool to hear all voices, not just those of us like me and John who are busy and loud in the ALS community, but the silent majority, the people we don't hear from, the people who aren't active on social media, but whose voice and whose interests and priorities matter to us. And so that is why I'm so excited about the focus survey. This is a chance to ask a series of recurring questions about priority issues and get systematic answers. So all of us in all of our organizations are more confident that when we say we're speaking on behalf of people with ALS and their caregivers, we actually are. So that's an invitation to you to take advantage of this tool of empowerment. It's up to us to be heard, and I encourage you both to participate in the surveys when they come out and to tell others to participate as well. Now, John and I have two asks of you. You can see this slide. The first is when this webinar is finished, not before, you have to wait for we're done, but when the webinar is finished, please go to the alsa.org/ALS Focus website and register for the forum for the focus survey. In July, we will have our second survey released, and at that point, we'll contact you and you can finish it. As you heard from Jill, there's two dimensions to the survey. The first one you only have to do once. It's the demographics. It's really important. It's boring to fill those out, but it's important because that gives us a chance to test some hypotheses about different variations in answer. We might find that on a particular issue, people with advanced ALS have a different preference than people in early onset, or people who are younger have a different preference than older. So that's a really important part of it. You only have to do it one time, and then you can get to the recurring question, or the one-off surveys that we'll ask each time, and those should only take about 10 minutes. We really welcome your participation. I hope we get a big turnout. Second, John and I are also interested in seeing our patient and caregiver advisory committee expanded. We've currently got 13 members, 9 people with ALS and 4 caregivers. Would like to expand that a bit. If you are interested in participating, please email to the email address listed on the PowerPoint slide. We promise we'll get back to you. We may not have room for everyone, but once we've got your name and your email address, we can certainly find opportunities for collaboration with you on a variety of patient-centric initiatives with the association and beyond. Thank you so much for your time and attention. I hope you're having a great day.
Ken, John, thank you so much. And John, if you want to jump on back on video, that'd be great if you want. Oh, there he is. So thank you both for taking time to help me today to talk about this amazing program. And it's just been awesome working with both of you guys and the community on this really important program. So Morgan, we're happy now to stop and take the last few minutes to take any questions around ALS Focus.
That would be terrific, and I have lots of them for you. Someone has asked if we include chapter care service coordinators in the survey program because they are such an excellent point of contact to our patients and their caregivers. To what extent do we involve our care service coordinators at Focus?
Sure, I can start with that. So we are always in touch with the care service leads across the country around keeping them up to date on what we're doing with ALS Focus. They're a huge part of helping us promote the program. And what we would like to do in the future is bring them more closely in to do some testing around promoting ALS Focus at clinics and at support groups across the country. And Ken and John could talk more about those efforts. But our care service leads across the country are super important in helping us get the word out. And they actually helped me find Ken and John and the members of our patient and caregiver advisory committee when we first started. So that was awesome too.
Great. And can you just elaborate a little bit more on how exactly patients and caregivers are involved and what they contribute to the ALS Focus patient and caregiver committee?
Ken or John, do you want to take that one since you run the committee? You're on mute.
Ken, you're going to need to unmute yourself.
I'm sorry. What I said, unfortunately, is that I didn't hear the question fully because we've got a storm and my link is not very good. Can you repeat the question?
Sure. I'm asking you guys to elaborate a little bit more on the role of patients and caregivers in the ALS focused program and on the PCAT. Sorry, I can hear the feedback.
I'm sorry, I can't hear you. John, can you take it?
That's all right, I'm going to push it over to John.
Yeah, don't blow away. So at the end of the day, the patient and caregiver committee is the group of people that voice our opinions, patients with ALS and our caregivers on all subject matter relative to focus surveys. So we actually help formulate questions on the surveys as well as choose survey topics amongst a larger group of people, including folks from the industry, the scientific world, and the FDA.
Yeah, and just to add, yeah, I don't do anything unless I talk to Ken and John, their committee, essentially. Everything from picking the survey topics to the questions to decisions how we're going to share data, how we're going to promote the program. Really every aspect has been informed by John and Ken and our team with the PCAT.
And can you explain, we've had a couple of questions asked for you to differentiate between the ALS registry and the ALS focused program. Which is which?
Ken, do you want to go or do you want me to go?
We're talking about the ALS National Registry, the CDC registry?
Mm-hmm.
Yes, what's the difference between the registry and
that's a totally different beast. The National Registry is intended to be a comprehensive repository of demographic and other data for every person with ALS who registers, which we're trying to make universal. You know, that's one of the challenges, is making sure everyone who has ALS actually registers for it. And that then provides researchers with a rich repository of data on all kinds of things related to exposure to pesticides, related to other environmental factors that eventually is going to be critical for researchers to determine what causes ALS, the various combination of factors that have been, you know, that have just befuddled us to date. So it's a really critical research tool. By contrast, our focus surveys are asking a far smaller set of demographic questions that we think are critical for helping us analyze our specific surveys. So they're two very different things.
Yeah, and what's cool is that we're able to, people who recently signed up for the ALS registry who have been assigned a global unique identifier will be able to connect their data to our data, which is very, very interesting to see how the questions we ask around patient really focus around patient experience and burden and the challenges you face, and connect them to what you've been exposed to in relation to environmental risk factors. Go ahead, John.
I would just like to add that the focus program is going to also be an avenue for the ALS Association and anyone nationally or even internationally in the scientific community. They'll have access to the data. But what I think is unique is with the ALS Association database, we are going to be proactive in doing things like changing regulatory review process, drug development, clinical trial design, all the things that I listed earlier. We're going to proactively go after as the association. So the response of patients is going to help us determine what is the most important and how we lead the ALS Association to be proactive in the areas that matter to us.
Exactly. Thanks, John.
Thanks, and go ahead, Jill. I'm sorry, cut you off.
No, go ahead, Morgan.
Well, I've asked our team to put the slide back up because we're getting a couple of requests for information about how people can register and where they can posit some more questions about getting involved in the patient caregiver community. So, folks, here it is. Please take time to write this down. We would love to hear from you. I have one more question for you that quite a few people have asked. Unfortunately, we're at a time where not all the data remains private. What are the steps that you take to ensure the absolute privacy and security of data that our patients share with us?
Ken, do you want to go? You're shaking your head.
No, I was looking at you. I was looking at you, and that's what you've been working on. So please, Jill.
Yeah, we've taken a lot of time. My ALS focus director, Sarah Pavanta, is really ultra sensitive about this. She talks to our Western IRB daily. We're constantly thinking about how we can keep your data confidential. We work very closely with Neurobank, who has a lot of policies and procedures in place around data sharing and confidentiality. And the global unique identifier is really important to that process of keeping your work of your information confidential. It's really like a code that's really cool, unique code that's you, that we can't tell in any way that it is you. So, and we don't, in our demographic information, we're not asking a lot of really personal questions that we could somehow, if we did a lot of digging, find you. It's I would say, I can never say 100% impossible, but like super really hard to do. So we take that confidentiality part really seriously, and that's not something that we would ever let happen.
I would also just quickly add, there are no personal addresses included in the demographics. It's basically the first 3 digits of a person's zip code. So if someone is in a witness protection program, let's say, you won't be found.
Thanks, John.
You've reassured a lot of people there, John. And by the way, the John Russo fan club is out in force. No, not a question, but a comment. I love John Russo. So there you go.
We all do.
And that's a great note to end this section of our program on. Dr. Yersak, John, Dr. Mankhouse, thank you very much for your time. I'm going to, turn our floor back over to Dr. Thacker.
Thank you, Morgan. And Ken and John, thank you for your leadership on this program. It's been really wonderful working with you guys and incredibly helpful to us. I also want to thank Dr. Benatar for his presentation. That's probably the best explanation of neurofilaments I've seen. It was really helpful. So thank you, Morgan. Thank you, Caldup and Jill as well. And thank you, our sponsors, Alexion, Amylyx, Biogen, Genentech, Mitsubishi Tanabe Pharma America, and the Association Pharma for making this possible. And thank all of you for listening. As you can see, there is a lot happening in the research space, a lot. It's very dynamic. And there's a whole bunch of different players that are involved in this. It's a complex interplay between the FDA and the NIH and the CDC and us and the VA and the Department of Defense. And that's why all of these things are really important, and it's really important Congress understands how important research is, how important all of these pieces are properly funded. And your work in the advocacy program is especially important. So thank you. And I'll close it here, and I look forward to talking with you more tomorrow.